Alrighty, so today, along with the online diffusion lab, you're going to be also taking some notes on cell movement. These notes are in the unit packet if you want to follow along there. If my page will change. So the membrane structure. So you've all learned about the plasma membrane already. It's one of the organelles, and the plasma membrane, it just regulates what goes in and out of a cell. It's the very outer layer of most cells that regulates what goes in and out. I'm going to go kind of fast through these, so if you need more time to write down, just go ahead and pause the video. So the membrane structure is composed of a phospholipid bilayer. And a phospholipid bilayer is just a two-layer sandwich that the molecules form around. So as you can see in this picture here, this is the phospholipid bilayer where all the heads are on the outside and all the tails here face inwards. Sorry about that. Miss Julius walked in and ruined my whole video. So back to the structure of a membrane. Again, it's a phospholipid bilayer. It has two parts. There's heads and tails to the phospholipid bilayer, which I'm going to explain in this side. So there are hydrophobic and hydrophilic parts of the bilayer. Hydrophobic, if you break that word down, the prefix hydro means water, and the word phobic means hating or fearing. So if you have arachnophobia, you're afraid of spiders. Hydrophobia means you are afraid of water. The tails that are in the middle of this phospholipid bilayer are hydrophobic. They don't want water anywhere near them. They do their best to stay away from water. Whereas the heads that are on the outside of the bilayer are hydrophilic. So again, hydro meaning water, and philic meaning loving. So they're water-loving heads. That hydrophobic and hydrophilic qualities are super important to the membrane because they allow certain things in and not other things in, which we're going to talk about in a little bit. There's also a lot of membranes embedded in the proteins, and there's a lot of proteins embedded in the membrane, and these proteins make channels so that things can get through. Because like I just said, the membrane is really good at allowing some things in and other things not in. So these proteins help make channels for the things to get in and out more easily. So general movement of molecules. So in a membrane, water, nutrients, gases, and ions are constantly moving in both directions, in and out of a membrane. And there's a lot of different ways to do so. Generally, everything will move with a concentration gradient. So what a concentration gradient is, it starts where something is really concentrated, really, really high, and then they're going to move to somewhere where the concentration is really low. So notice in this first picture, there's a bunch of molecules inside the cell and not a lot outside. The goal of this is always to reach what we call equilibrium, which is an equal amount on both sides. So these particles that are inside the cell are going to move out from an area of high concentration to low. Whereas here in this picture B, they're going to move from the outside, where there's a lot of particles, to the inside, where it's low. This is what we call diffusion. So diffusion is something moving down its concentration gradient from an area of really high concentration to an area of really low concentration. So you can think about diffusion like when someone farts. If you're right next to the person who farts, you smell the fart first. But if you're a little bit away from the person who farts, it takes you longer to smell the fart because the particles from the toot take a long time to disperse around the room. They go from an area of high concentration, where the person farted, to an area of low concentration, where the person is not located. So again, diffusion is just things moving across the membrane from an area of high concentration to an area of low concentration. And you should have saw that in your lab you just completed online. Ideally, a diffusion will never stop. Things will move randomly until they reach equilibrium. And like I said, equilibrium is just when there's equal amounts on both sides. However, when things reach equal amounts on both sides, things just don't freeze like that. Things keep moving back and forth, but they move it back and forth in equal amounts. So if one particle goes this way, one goes this way. If three go this way, three go this way. And this keeps happening. So we never have standstill motion in a cell. It's constantly moving, but in equal amounts to keep that equilibrium. And again, equilibrium is the number of molecules moving in one direction is equal to the molecules moving the other. There's a balance there. So I mentioned earlier that the membrane allows some things in and other things not in. That term for that is that the membrane is selectively permeable. It straight up means that some things are allowed to cross the membrane and some things aren't. Some things can get across easily and some have more trouble. It's selectively permeable to different types of materials. And you should have learned that in that cell movement POGIL that you did, I believe, last week on Friday. Or like most of you do on Sunday night. So there's some other different types of transport. So in general, there's what's called passive transport. Passive transport means that no energy is required. It just happens automatically. So diffusion is an example of passive transport. Passive transport usually involves small particles because small things are easy to have an easier time crossing a membrane. There's also what's called facilitated diffusion. So I said general diffusion is just when things go from high concentration to low concentration with no energy required. But there's also what's called facilitated diffusion. The word facilitate means to help or assist. So facilitated diffusion is the fact that proteins are helping make a path for molecules to pass. So as you can see here, we have our phospholipid bilayer, and we have those proteins that I talked about that were embedded in that bilayer. These proteins can help provide a path for different molecules to pass. So if molecules can't get through the membrane on their own, they might go through this protein so they can get from one side of the cell to the other. Again, in facilitated diffusion, things are still going from concentration gradient of high to low. Another type of movement is what's called osmosis. Osmosis is just the movement of water. Diffusion is the movement of anything. Osmosis is specifically the movement of water across a membrane. And of course, that water is going to go from a concentration of high to low. Another type of transport is called active transport. So we talked about passive transport, it just happens. But active transport requires an input of energy to move something across a membrane. The reason it's requiring energy is because something is moving against its concentration gradient. It's going from an area of low to high. Going from high to low is easy. The molecules just want to go there. But if you want to push something the other way, you have to use energy to do it. And then our last two types of movement of molecules we're going to talk about is endocytosis. Endocytosis is taking materials into a cell. So cyto means cell, endo means in, so we're taking things into the cell. Whereas exocytosis is we're exiting things. Things are exiting the cell. Things are moving out of the cell. If you have any questions on these notes, please let me know.